the estuaries of the eastern United States, the water carries a chemical threat that can deform a fish before it hatches. Dioxin-like PCBs and polycyclic aromatic hydrocarbons collect in contaminated sediments. In sensitive Atlantic killifish embryos, they can trigger severe heart defects and death. Yet some killifish still live in places such as New Bedford Harbor, Massachusetts, and Newark Bay. Their advantage is small enough to hide inside a cell. Changes in the aryl hydrocarbon receptor pathway reduce the usual cascade of toxic responses. The fish does not become invulnerable. It becomes less damaged by a particular poison. In laboratory comparisons, adapted populations tolerated exposures that killed embryos from cleaner populations. In some tests, the difference reached 2 or 3 orders of magnitude, 100-fold to 1,000-fold separation, like surviving a dose measured in drops, while another population faces the same chemical by the cupful. What happens when a fish evolves to remain where other fish disappear? Does its survival preserve the old food web or create a new one? Why do unrelated crustaceans keep arriving at the shape of a crab? And when the ocean rewards an animal for becoming enormous, what happens when the feast moves? You're watching Beyond the Familiar. Join us as we follow one adaptation from a poisoned estuary into ancient predator-prey battles. Watch the crab body plan evolve more than once, and then enter the productive currents that made ocean giants possible, before asking what happens when yesterday's solution meets tomorrow's sea. The first rule is simple. Evolution does not begin with improvement. It begins with pressure. A population contains variation. The environment filters that variation through survival and reproduction. If a heritable trait changes the odds, its genetic basis can become more common. No organism needs to foresee the problem. No lineage needs to be moving towards perfection. The Atlantic killifish shows this process in a modern setting. At contaminated American estuaries, industrial pollutants became a persistent selection pressure. The aryl hydrocarbon receptor, or AHR, normally helps cells respond to certain chemicals. In polluted killifish, that response became profoundly desensitized. CYP1 enzymes, which are often strongly induced by these compounds, were also altered. Whitehead and colleagues documented the pattern in studies of polluted killifish populations, including work published in 2012. Their genomic and physiological results found similar AHR pathway changes in several geographically separate populations. The same broad solution appeared more than once. That matters because the fish didn't merely become temporarily accustomed to dirty water. When offspring from polluted populations were raised for 1 or more generations in clean laboratory conditions, their resistance persisted. A response that survives the removal of the original environment points towards heredity rather than short-term acclimation. A later genome-wide study reported at least 8 regions showing elevated differentiation between polluted and reference populations. Candidate genes were associated with AHR signaling, detoxification, and cardiac structure and function. One quantitative trait analysis attributed about 69% of variation in tolerance to mapped genetic regions. The AHR2 region contributed about 17%, while additional regions and interactions accounted for much of the remainder. The arithmetic isn't a magic gene. It's a reminder that adaptation can be distributed across a population's genome. So, what did the killifish gain? It gained access to an estuary that toxic exposure had effectively closed to sensitive fish. That can preserve a small-bodied fish in the local food web, maintaining a link between polluted sediments, invertebrate prey, and larger predators. The evidence for that community-scale effect is more limited than the evidence for genetic resistance, so we should keep the verbs honest. The killifish adaptation clearly protects the population, It appears to help preserve a trophic connection. But the advantage carries a bill. Field populations can still show high rates of liver tumours. Altered AHR signaling may produce trade-offs elsewhere in the body. The fish hasn't escaped environmental stress. It has shifted which stress it can tolerate. One pressure, one cellular adjustment, one altered population. Then the consequences widen. But here's where it gets interesting. The same kind of adaptation can appear independently when separate populations face similar demands, even though their histories aren't identical. In Atlantic cod, intense fishing pressure during the 20th century caused abundance crashes and a shift towards earlier maturation in populations on both sides of the Atlantic. In 2023, Brendan Reed, Bastian Starr, and Marlin Pinsky used temporal genomic data from cod sampled in Canada and Norway. The dataset contained 346,290 called single nucleotide polymorphisms, tiny DNA differences, across samples spanning roughly a century. Their analysis in Philosophical Transactions of the Royal Society B found genome-wide covariance in allele frequency change consistent with parallel polygenic adaptation. The word polygenic means that many genetic locations contribute small effects. Think less of a single switch and more of a crowded control panel with dozens of sliders moving in the same direction. The cod study also made an important distinction. Similar life history shifts can result from plasticity, where individuals change during their lifetime, or from evolution, where inherited variants become more common. Temporal genomic patterns can help separate those possibilities, but they don't make every observed change automatically evolutionary. This is the discipline we need for the ocean's deeper history. A trait may be useful, a predator may be present, shells may become thicker, but the chain from pressure to trait to ecosystem change must be demonstrated, not merely imagined. The fossil record preserves some chains beautifully. It also leaves gaps. In the Cambrian Ocean, approximately 517 million years ago, what is now South Australia lay beneath seawater filled with small shelled animals. Among them was Lapworthella fasciculata, a tomateid related to brachiopods. Its body was protected by separate mineralized pieces called sclerites. A study published in Current Biology in 2025 examined more than 200 of those sclerites. Many contained holes attributed to a perforating predator. Through the sampled sequence, perforations became more frequent. At the same time, the sclerites became thicker, and perforation frequency correlated positively with thickness. The picture is stark. A predator reaches through the armor. The prey population contains variation in shell thickness. Thicker pieces become more common as attacks continue. The predator still gets access often enough to leave more holes. This is the oldest empirical fossil evidence described for a microevolutionary predator-prey arms race. But the fossil does not give us the predator's name. It does not show the attacker's mouthparts changing generation by generation. It records repeated damage and a prey morphology trend. The prey defense is strongly supported. Full reciprocal evolution in the predator remains partly inferred. So, is this a complete two-sided arms race? Not quite. It's stronger to say that the evidence shows continued predation alongside a shell-thickening response. The missing predator anatomy matters because coevolution requires reciprocity. One species changes in response to another, and the other changes in return. That distinction becomes clearer in a living shoreline. The European green crab, Carcinus maenas, expanded into northern New England around 1900. On the rocks and mudflats lived the flat periwinkle, Littorina obtusata, a snail with a shell whose shape could be compared across historical collections. Specimens gathered before the crab's expansion had high-spired, thin-walled shells. Specimens collected in 1982 to 1984 had lower-spired, markedly thicker-walled shells. The shell had become harder to crush. In 2004, a PNAS study reported that snails exposed to waterborne cues from predatory crabs rapidly produced thicker shells than control animals. The crab didn't need to be seen. Chemicals in the water were enough to signal danger. What does a thicker shell cost? Increased shell production is associated with reduced body mass, reduced growth, or both. Resources invested in carbonate walls can't also be invested in soft tissue. The snail has exchanged speed of growth for resistance at the crusher's claw. The response is partly plastic. A snail can alter its shell during its lifetime when crab risk rises. Yet the geographic pattern also points towards selection. In 2024, researchers found that southern populations with longer exposure to green crabs tended to pay a lower tissue mass cost while producing thicker shells than northern populations. The southern snails weren't merely wearing heavier armour. Their populations appeared better adjusted to the price of making it. What option did the crab close? The thin shell strategy. Grow with less investment in protection, remain exposed, and spend more energy on tissue. Once a shell-crushing predator becomes common, that option becomes dangerous. The snail opens another route through reinforcement, but the route narrows its growth budget. This is a real ecological response, but the supplied evidence documents a one-sided prey defense example rather than a completed reciprocal genetic arms race. The supplied evidence does not show that green crab claws or feeding behavior evolved in response to the thicker shells. A famous summary of the broader pattern, reproduced in a review of Geert Vermeer's work, described how thickness and spinosity of snails matched with growing strength in the claws of crab predators. It's a vivid phrase. It's also a synthesis of a broad fossil pattern, not a laboratory record of one crab species and one snail species moving step by step together. Vermeer introduced the Mesozoic marine revolution concept in 1977 to describe an escalation in marine predation and prey defense. Shell-crushing predators diversified across groups, including teleost fishes, rays, heterodontid sharks, stomatopods, lobsters, and crabs. Their tools differed: crushing, drilling, puncturing, and forcing open. Prey responded with thicker shells, ribs, spines, reinforced apertures, deeper burrowing, and greater mobility. The important unit isn't "predator gets better, prey gets better" as a slogan. It's access. A thin, exposed animal is accessible from above. A burrowing animal isn't in the same layer of the sea floor. A swimming scallop isn't offering the same target as a stationary bivalve. A squid can leave the sediment entirely. One solution closes a habitat option. Another solution opens a different one. By the middle Mesozoic, fossil assemblages preserve abundant damaged and crushed shells, including accumulations in Middle Jurassic clays of the Polish Jura. At the same time, increasing infaunality, life buried within sediment, helped create more vertically tiered benthic communities. The seafloor became not one flat surface, but a stack of living spaces. The evidence does not support a smooth, uninterrupted escalation. Most shell-crushing predators were probably generalists, so assigning a particular defense to one named predator is often impossible. Long intervals of relative stability were punctuated by abrupt reorganizations after mass extinctions. The arms race wasn't a staircase. It was a landscape of pressure, pause, extinction, and renewal. But the revelations don't stop there. The same logic appears in open water, where the armor is microscopic and the battlefield is a mouthful of plankton. Diatoms are photosynthetic organisms enclosed in silica-rich frustules. Copepods graze on them, but heavily silicified diatoms can be mechanically difficult to break. Some diatoms also produce toxins, alter colony size, or form rapidly sinking aggregates that reduce grazer access. The copepod isn't passive. Feeding structures can experience wear while processing defended prey, and copepods show selective feeding behavior alongside adaptations that help them handle diatom toxins and mechanical resistance. This modern interaction has been described as an evolutionary arms race involving both feeding and defense. It may influence plankton diversity and the biological carbon pump, the chain of processes that moves carbon from surface waters into the deep ocean. That larger effect is an interpretation supported by the ecological role of both groups, not a fully reconstructed geological sequence for every lineage. Still, the mechanism is visible. A microscopic shell changes what a grazer can eat. Feeding selectivity changes which cells remain. Sinking particles change where carbon travels. At this scale, a trait can redirect the ocean without ever becoming large enough to see. So, what's the truth about adaptation? It isn't an upgrade installed in one organism. It's a change in the menu of possibilities around that organism. A shell can make a snail safer but slower growing. A burrow can protect prey but remove it from surface-feeding communities. A diatom's silica can hinder a copepod while altering the movement of carbon. And sometimes the solution escapes the original problem entirely. The word carcinization was coined by the zoologist Lancelot Alexander Borrowdale in 1916. It describes the repeated evolution of a general crab-like body form in crustacean lineages that weren't originally crabs. The recurring features include a broadened and flattened carapace, a compact body, a reduced abdomen folded beneath the animal, a reinforced exoskeleton, and reorganized locomotion. In a shallow coastal world of rocks, crevices, sediment, and waves, a low, compact body can improve stability and movement across the sea floor. A rigid shell can assist protection and grasping. Strong claws can help with feeding and defense. These are functional interpretations, not proof that every crab shape evolved for exactly the same reason. Why do unrelated animals keep arriving at a crab-like outline? Because similar environments can filter towards similar working shapes. That is convergence. True crabs, the Brachyura, aren't the same evolutionary lineage as porcelain crabs, king crabs, or the hairy stone crab, Lomis hirta. King crabs belong to Anamuran crustaceans, a group that also includes hermit crab lineages. Their crab-like appearance wasn't simply inherited from true crabs. The distinction is crucial. The basic decapod framework, segmentation, a carapace, 5 pairs of walking legs, and abdominal segments, comes from common decapod ancestry. The specific crab-like modifications evolved independently. In 2013, Bracken Grissom and colleagues used an integrative reconstruction of Anamura and inferred 3 independent crab-like origins within that group, in the lineage containing king crabs, in porcelain crabs, and in Lomicidae. In 2017, Kyler, Werkner, and Richter reviewed a century of carcinization research and reached the same broad conclusion. Crab-like habitus evolved at least 3 times independently within Anamura, separately from true crabs. Other analyses report different counts when they include multiple true crab origins, partial crab-like forms, secondary losses, and later re-evolution. Under broader definitions, at least 5 major independent crab-like lineages are reported. The exact number depends on what counts as a separate origin. Evolutionary history refuses to provide a tidy scoreboard. What the evidence does support is more interesting than a fixed total. A crab-like body plan isn't an inevitable destination. It's a shape that can be reached, abandoned, and reached again. McLaughlin and Lemaitre's 1997 study, Carcinization in the Anomura, Fact or Fiction? 1. Evidence from adult morphology challenged the simple idea that king crabs evolved directly from hermit crabs through one straightforward transformation. Externally similar animals can have different internal anatomy and different evolutionary routes. The crab shape is a useful analogy, only until we start treating outline as ancestry. A flat body does not reveal a shared origin. A folded abdomen does not tell us the developmental path that produced it. Resemblance is evidence of similar form, not automatically evidence of close relatedness. And the story has a reverse. Decarcinization describes the secondary loss of a crab-like condition. Multiple living and fossil lineages became more cylindrical or long-bodied after evolving crab-like forms. The shape that was favored under one set of pressures could become less useful under another. A body plan can be an answer without being a final answer. A previous evolutionary conversation popularized the question in the phrase, "Why do animals keep evolving into crabs?" That title captures a genuine scientific puzzle, but its audience appeal isn't evidence for the number of origins. The evidence comes from morphology, molecular phylogenies, and comparative evolutionary studies. The editorial hook points us toward the question. The ancestry tests decide the answer. But that's not even the strangest part. Once the body plan becomes successful, evolution can change not only its shape, but its scale. For much of their early history, baleen whales were small to moderate in size. The modern blue whale can reach approximately 30 meters, about the length of a 10-story building laid on its side. But that extreme didn't appear when baleen whales first evolved. A clade-wide shift towards very large body sizes occurred during approximately the last 4.5 million years. Fossil and paleoceanographic evidence links the shift to stronger seasonal upwelling, glacial delivery of nutrients, and dense, patchy prey concentrations such as krill. The shift toward enormous size has been linked to an ocean offering food in concentrated places and seasons. That is one explanation. It isn't the explanation for all marine gigantism. In water, buoyancy removes some of the structural limits that constrain land animals. Cold water can contain more dissolved oxygen, and low temperatures can reduce metabolic rates. But the oxygen temperature hypothesis isn't universal. Woods and Moran's 2020 review in Integrative and Comparative Biology found that evidence differs among organisms, partly because ventilation and internal oxygen transport can matter as much as passive diffusion. A 2019 study by John Spicer and Simon Morley tested Antarctic amphipods across a wide range of body sizes. Their results found that larger body sizes were generally more sensitive to declining oxygen, but species identity also mattered. One more active intermediate-sized species performed better than the oxygen limitation hypothesis alone predicted. The ocean does not offer one universal giant-making recipe. Productivity, prey abundance, temperature, oxygen, buoyancy, migration, physiological design, habitat, and ecological opportunity interact differently in different lineages. A giant polar amphipod and a giant baleen whale aren't solving the same problem. Marine mammal data show a particularly strong connection between large size and productivity. In 2016 and 2017, studies linked increasing body size in marine mammals to productive regions with dense, often seasonal prey patches. Baleen whales can exploit these concentrations through bulk filter feeding. The advantage of a large body isn't simply that it can eat more. It can travel between distant feeding and breeding areas, store energy between meals, and process enormous volumes of water when prey gather densely. But size changes the food web around the animal. A 30-meter filter feeder does not forage like a smaller predator taking individual prey. It follows seasonal productivity, often moving across ocean basins. The landscape becomes part of its feeding strategy. Large whales also move nutrients. The Marine Mammal Commission describes the whale pump, in which excreted nitrogen and other nutrients return to surface waters and support phytoplankton growth. When a whale dies, its carcass can sink as a whale fall, carrying organic matter and carbon into the deep sea. The animal is therefore not only consuming the food web, it helps transport material through it. The fossil record records a related community shift. As very large mysticetes became more prominent, several smaller forms, including Bailey nula, Bailey nella, herpetocetines, and small balaenopterids declined or disappeared. Did gigantism alone eliminate them? No. Productivity shifts, climate, competition, habitat structure, and the incompleteness of the fossil record all complicate that conclusion. The evidence supports a body-size transition associated with new oceanographic conditions. It does not prove that becoming large directly caused every smaller lineage to vanish. Other marine giants show why context matters. The fish Leadsichthys problematicus has been estimated at approximately 16.5 meters, roughly the length of a city bus and a half. The crinoid Cyrocrinus subangularis reached approximately 15 meters in vertical living structure. The straight-shelled cephalopod Endocerus giganteum reached approximately 9 meters. A study of Paleozoic marine invertebrates found an average increase in extinct marine body size of about 2.2% per million years, with a 95% confidence interval from 0.6 to 3.8%. Yet it found no straightforward correlation between maximum size and oxygen, temperature, or sea level. The signal was strongest where clade diversity was high. That suggests ecological opportunity mattered. More kinds of organisms can mean more roles to occupy, more prey types, more habitats, and more ways for large bodies to pay their energetic costs. Still, size is never free. A large animal needs more energy in absolute terms. Its generation time is longer. Its population can recover more slowly from losses. If prey patches shift or disappear, a body built for high-volume feeding becomes a costly machine waiting for fuel. The same body that crosses an ocean efficiently may be unable to survive a long gap between productive feeding grounds. The same specialization that makes feeding efficient in a predictable prey field may narrow the alternatives when that field moves. So what happens when success creates dependence? We can answer that with stronger evidence for vulnerability than for a single dramatic extinction story. The fin whale provides a cautious modern example. In 2024, a study reported declines in individual and population dietary specialization during an ecosystem shift, accompanied by lower recruitment and survival. The whales weren't simply choosing a new menu in a stable world. Their feeding patterns changed as the ecosystem changed around them. The result is consistent with a specialist-prey mismatch. It does not prove that dietary specialization alone caused the decline. Prey redistribution, climate, competition, and demographic effects may all contribute. That uncertainty isn't a weakness in the story, it's the mechanism we need to respect. Specialization can be efficient when a resource is abundant and predictable. It becomes risky when the resource moves faster than the animal can follow, or when the habitat that supports it disappears. The pattern appears across reef fishes as well. In 2021, Rick Stewart-Smith, Camille Mellin, Amanda Bates, and Graham Edgar analyzed the Global Reef Life Survey dataset covering 4,070 transects. After regional coral mortality and between sea urchin barrens and kelp habitats, communities shifted consistently towards fishes with broader habitat requirements. Generalist tropical reef fishes also extended farther into subtropical and temperate zones than specialists. A specialist isn't necessarily poorly adapted. It may be exquisitely adapted to one habitat. That is precisely the danger. When coral structure is lost, a fish dependent on particular shelter or food can't simply substitute any nearby habitat. A generalist can occupy more options. The specialist may remain alive while losing the places where its life cycle works. The sea changes first as a physical system, then as a map of opportunities. A warming current shifts prey north. A bleaching event removes coral architecture. A productive upwelling weakens. A pollutant excludes sensitive competitors and leaves a resistant fish behind. Each change alters who can feed, hide, reproduce, or migrate. Marine extinction records demand similar caution. A 2023 review by Pablo Del Monte Luna and colleagues found between 20 and 24 documented marine species extinctions over the previous 500 years, while population losses were estimated to occur 2 to 10 times faster than 5 centuries ago. For the ocean, disappearance is difficult to detect because empty water does not announce its last survivor. The clearest modern pattern isn't one trait causing one extinction, it's interacting pressure, exploitation, habitat change, climate, bycatch, prey decline, warming, and oxygen loss. Large body size, restricted range, low reproductive rate, and specialized diet can combine into vulnerability. None alone provides a complete explanation. The title calls these evolutionary accidents that change the ocean forever. If we ask a scientist to define accident, that word is media shorthand rather than a scientific category. Genetic variation isn't directed towards a known future. Natural selection filters what already exists against the conditions that happen to prevail. An unusual shell, a desensitized receptor, a flattened body, or a larger frame can spread because it works here, now. Forever is also a poetic scale, not a literal endpoint. Cambrian shells changed predator access. Mesozoic defenses changed the architecture of the sea floor. Crab-like bodies appeared repeatedly. Marine gigantism reorganized feeding and nutrient movement. Each effect persisted beyond the first individuals that carried the trait. But no ocean stopped evolving. The transformation continued. That is the real chain. Pressure makes a trait valuable. The trait changes access. Access changes behavior. Behavior changes selection. The new solution creates a new cost. When the environment shifts, the cost may become the first thing we notice. We can see the chain in a killifish embryo surviving a toxin that once killed it. We can see it in a snail trading tissue growth for a thicker shell. We can infer it from a Cambrian sclerite pierced more often as it thickens. We can trace it through crab-like bodies that evolved independently, then sometimes disappeared. We can measure its largest expression in whales following seasonal rivers of krill. And we can see the warning in the gaps. We don't know every predator behind every fossil hole. We can't reconstruct every ancient feeding decision. The exact count of crab-like origins changes with phylogeny and definition. Gigantism has no single cause. No supplied evidence proves one specialized marine lineage went extinct solely because of one evolved trait. The ocean isn't a machine with one lever. It's a negotiation among thousands of solutions, each one altering the conditions faced by the others. The final image isn't a crab, a whale, or a shell on its own. It's a changing seafloor beneath a moving surface, burrows opening where exposed life could no longer remain, plankton sinking with carbon in their bodies, a whale crossing between distant feeding grounds, and a small fish surviving in poisoned water because its cells learned not to answer the poisons so loudly. These representative solutions leave shapes in the ecosystem. Some shapes last for millions of years. Some collapse when temperature, oxygen, predators or prey move beyond the range that made them useful. Adaptation is powerful but provisional. It does not promise progress. It gives life another option, and that option changes the world around it. My friends, the sea we inherit is built from old answers to old pressures. The question is, which answers still leave room for what comes next? Which adaptation do you think most dramatically redirected an ocean ecosystem? The shell, the crab-like body, the giant filter feeder, or the microscopic defense? Share your choice and the evidence that persuaded you. I'll see you beyond the familiar.